Hello, my name is Chelsea Fountain, and welcome to my final group advanced concept presentation. We've been working on this presentation since week 4 leading up to week 7, and this is my end result of my presentation poster board. I will walk you through this evening the process of how nucleotides and ribose are made and show how these come together to make ATP used for DNA replication. So let's begin with the pathway of building glycolysis. Glucose, we will start with this by looking at images 7, 2, and 8 as shown on the poster board. So glucose is the principal carbohydrate in living systems and an important fuel. Glucose is one of several monosaccharides formed from formaldehyde and the most stable hexose because the hydroxyl groups and hydroxymethyl groups. As you can see on images 7, 2, and 8, the glycolytic pathway can be divided into 2 stages. Stage 1, glucose is trapped, destabilized, and cleared into 2 interconvertible 3-carbon molecules generated by cleaved of 6-carbon fructose, and no ATP is generated at this stage. Stage 1 also begins with the conversion of glucose into fructose 1,6-biphosphate, which consists of 3 steps: phosphorylation, isomerization, and second phosphorylation reaction. In stage 2, the 3 carbon units are oxidized to pyruvate, generating ATP. Remember, the hexokinase trapped in glucose in the cell and begins glycolysis. Glucose enters cells through specific transport proteins and has one principal fate inside the cell. It is phosphorylated by ATP to form glucose 6-phosphate. The transfer of the phosphoric group and ATP to the hydroxyl group on carbon 6 of glucose is catalyzed by hexokinase. The glycolytic pathway is tightly controlled and has a dual role that degrades glucose to generate ATP, and it provides building blocks for biosynthetic reactions such as the formation of fatty acids and amino acids. The pathway, as shown on this image of gluconeogenesis, is located in the cytoplasm except for pyruvate carboxylase in the mitochondria and glucose 6-phosphate membrane bound in the endoplasmic reticulum. The entry points are for the lactate, the glycerol, and amino acids. Next, we move forward to the citric acid cycle. As you can see, we completed in the images 13, 12, 11, which you will see the citric acid as outlined on my poster, the electron transport train, the mechanism of ATP synthesis. So the citric acid cycle is the central metabolic hub of the cell, and it consists of 2 stages. 1, condensation of the acetyl coenzyme A with oxaloacetate, followed by 2 oxidative decarboxylations. Oxaloacetate is regenerated, coupled with the formation of high transfer protein electrons in a molecule of ATP. It is the way to the aerobic metabolism of all fuel molecules. The cycle is crucial for anabolism, which serves as an important source precursors of the building blocks of amino acid nucleotide bases in excuse me, I do struggle with some of these words. The citric acid is a component of the oxalate is another important precursor to the glucose. The synthesis of the acetyl coenzyme A by the pyruvate dehydrogenase complex is a key irreversible step in the metabolism of the glucose. The citric acid cycle is regulated primarily by the concentrations of ATP and NADH. The key control points are the enzymes isocytrate dehydrogenase and A-ketoglutarate dehydrogenase. The components of the electron chain, the components of the electron transport chain, the electrons flow down an energy gradient from NADH to O2. The flow is catalyzed by 4 protein complexes. The electron chain has high energy of electrons in the form of NADH and FADH2, which are generated by the citric cycle. Overall, the flow of the electrons through complexes 1, 3, and 4 of the electron transport chain leads to the transfer of proteins from the matrix side to the cytoplasmic side of the inner mitochondrial membrane. Next, we would then progress to the pentose phosphate pathway, which is metabolized by glucose 6-phosphate, which is you will see outlined in images 1 and 3 of the poster board. The pentose pathway is present in all organisms, which generates the NADPH and the ribose 5-phosphate in the cytoplasm. The pathway starts with the dehydrogenation of glucose 6-phosphate to form a lactone, which hydrolyzes to give 6-phosphogluconate and then oxidatively decarboxylated to yield ribose 5-phosphate. Lastly, as you just see all of the remaining images of the poster board, we'll discuss the nucleic acids consisting of bases linked to sugar phosphate. Backbone DNA and RNA are linear polymers of a limited number of monomers. In DNA, the repeating units are nucleotides, with the sugar being deoxyribose and the bases being adenine, thymine, glutamine, and cytosine, whereas in RNA, the sugar ribose in the base uracil is used in place of thymine. Overall, DNA can adopt multiple forms and be associated with specific proteins, whereas RNA can adopt and elaborate structures. I do hope you enjoyed this presentation of how nucleotides and ribose are made and show how these come together to make ATP used for DNA replication. And please excuse my mispronunciation of some of these words. Thank you.